Hey guys, welcome back. Today I'll be discussing about the topic mechanism of concentration of filtrate. This is from class 11 biology. Let's start with today's topic. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta play a significant role in this mechanism. This is Henle's loop and here is vasa recta. Vasa recta is a minute vessel from the network of peritubular capillaries that run parallel to the loop of Henle. So these two structures play significant role in the mechanism of concentration of filtrate. Let's discuss how it is happening. The flow of filtrate in the two limbs of Henle's loop is in opposite directions and thus forms a counter current. Yes, the flow of filtrate here in descending limb is downwards and in ascending limb it is upwards. Thus it is forming a counter current. Now, similarly, the flow of blood through the two limbs of vasa recta is also in a counter current pattern. You can see here the flow of blood in descending limb of vasa recta is downwards and in ascending limb it is upwards. Thus, it is also forming a counter current pattern. The proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining an increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmolarity per liter in the cortex to about 1200 milliosmolarity per liter in the inner medulla. That can be seen here. This is 300 milliosmolarity per liter in the cortex region and here is 1200 milliosmolarity per liter in the inner medulla region. This increasing osmolarity is because of the proximity between the limbs of loop of Henle and the limbs of vasa recta and also the counter current patterns seen in these two structures. Okay, now this gradient is mainly caused by sodium chloride and urea. Sodium chloride is transported by ascending limb of Henle's loop which is exchanged with the descending limb of vasa recta. It can be seen here. Sodium chloride is transported by ascending limb of loop of Henle. This sodium chloride is exchanged with the descending limb of vasa recta. Okay. Now, similarly, NaCl is returned to the interstitium by the ascending portion of the vasa recta. You can see NaCl is returned to the interstitium by ascending portion of vasa recta. Similarly, small amounts of urea enter the thin segment of ascending limb of loop of Henle which is transported back to the interstitium by the collecting tubule. Here it can be understood small amounts of urea enters this ascending limb of loop of Henle, then it is transferred in this direction and gets returned back to the interstitium from the collecting duct. So this is how the gradients are caused by sodium chloride and urea. The above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the counter current mechanism. This mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate, that is urine. Human kidneys can produce urine nearly 4 times concentrated than the initial filtrate formed. Important for exams. I repeat this once again. Human kidneys can produce urine nearly 4 times concentrated than the initial filtrate formed. This is all about today's session. Let's discuss some important questions from the topic that we have discussed today. First question: maintaining an increasing osmolarity towards the inner medullary interstitium is mainly caused by? The answer is sodium chloride and urea. Next question: human kidneys can produce urine nearly dash times concentrated than the initial filtrate formed. Answer is 4.